Hello, everyone, and welcome to Science on the Sound with the Mass Center Aquarium. And so today we've got some really fun things to talk about. So I don't want to hold us up too long. So we're talking about plankton of Poverty Bay. You might be thinking, okay, plankton, not so exciting, but it really is, especially because we've got Emily Bjornsgaard here, who is our lead aquarist, but also if you volunteer here or been around a while, is also leading our harmful algal blooms sampling with sound toxins. And so we've been doing that for about a year now. So she's got some awesome pictures that she wants to show all of us and then talk about, the importance of plankton. Even though it's small, it is mighty and plays an important role in our ecosystem. So if you have any questions about plankton while we get talking, please feel free to enter them into the chat box. And if you've got questions that you want to save to the end, we'll also have some time for questions and answer at the end of this talk. But no further ado, we've got Emily Bjornsgaard here to talk about plankton of Poverty Bay.
All right. Well, welcome everyone. Thanks for having me today to talk about Plankton of Poverty Bay. My name is Emily Bjornsgaard. I am the lead aquarist here at the Mass Center. So day to day, I'm taking care of all the larger animals in our 3,000-gallon aquarium system. Though once a week, I am sampling for phytoplankton in our surface waters here just off the dock in partnership with Sound Toxins. Sound Toxins is an organization that works on Puget Sound plankton, so here in the inner Washington state. But we'll talk a little bit more about the organization in a little bit. I have been here at the Mass Center doing Sound Toxins work for about a year, like Ariel said. But my first phytoplankton experience was in 2018 when I was a summer volunteer for ORHAB, which is the Outer Coast Harmful Algal Bloom monitoring organization. So there were a couple different sites in Grays Harbor County where I would go out and take plankton samples of the water and then learned how to identify them on the microscope. These photos here are just a few of what's to come. All of the pictures of plankton that are in this presentation have been taken by me here at the Mass Center in the last year. So all very, very much plankton of Poverty Bay. But if you're joining us from maybe not the Des Moines area, you may be wondering, where is Poverty Bay? So Poverty Bay is part of the Puget Sound, central Puget Sound, right where these red arrows are. And it is in between Tacoma and Seattle. So we're right kind of on the water in Des Moines, right along Federal Way. And then Maury Island is the other side of Poverty Bay. So we'll be talking about the 5 W's of plankton. The 5 W's is a journalism concept that we want to cover all the essential parts of our story here. So we're going to talk about what is plankton. We'll talk about where and when is plankton found, who's doing the monitoring for plankton, and why do we care? And then at the end, we'll also talk about how, if you're interested, you can get involved with learning about plankton some more. And also a note about names. So all living organisms have at least one name, but it may not be the name that you're familiar with. A lot of species that we see on our beaches or in aquariums have common names. So on here, we have the modest clown dorid is a common name for the sea slug. Other areas, they might have different common names that they're using, but all living organisms have a scientific name that is used across languages, and it is a way to streamline what species you might be talking about if you're talking at a conference in another country. So most of our plankton that we'll be talking about today do not have well-known common names, but they all have scientific names. So those will be what we'll be using today. And scientific names are written in italics typically. So if you see an italicized name on here, that would be the scientific name. If you see SP or SPP, that means species. That means I haven't identified it all the way to the species level, but I have identified it to the genus level, or the kind of general group that plankton is in. So what is plankton? That is a very big question. So plankton translates from Greek meaning drifter or wanderer, basically anything that lives in the water that doesn't swim against currents. So it may have some forward swimming ability like a jellyfish. You can see them in aquariums. They do pulse in one direction, but they aren't going to be able to swim against a current. And a lot of the plankton we'll be talking about today is very, very small. So we're talking about the small end of plankton, things you need a microscope to see. Of course, there are, like the jellies, larger things that are plankton that you don't need a microscope, but we're going to focus on the really, really tiny things. Um, Traditionally, we think about two different types of plankton. We think of our phytoplankton and our zooplankton. So phytoplankton is a plant-like plankton. I say plant-like because they're not always plants, but they are photosynthetic. So they're taking in carbon dioxide, nutrients, and sunlight and producing oxygen. And our phytoplankton are typically single-celled. Zooplankton are considered animal-like. They can be single-celled or they can be multicellular, and they are usually considered grazers. So they are not taking in carbon dioxide and producing sunlight. They are eating those phytoplankton and other smaller organisms to get energy. However, when you zoom in on the plankton, both physically and conceptually, there is a lot of in-between species that aren't just phytoplankton and aren't just zooplankton. A lot of the phytoplankton we're looking at today fall into kingdom Protista. So this is a catch-all group that are not animals, not plants, and not fungi. So anything else that is also not bacteria kind of falls into kingdom Protista. And so we have our phytoplankton, our photosynthesizers, our zooplankton, our grazers. And then in 2019, a term was coined mixoplankton. So these are organisms that can photosynthesize and graze. So they're kind of the in-between species. And there's a lot of new research being done on this group that kind of doesn't fall into either of the standard plankton types. So the three different types that we're going to be talking about today mostly will be diatoms, dinoflagellates, and larger zooplankton. And so jumping right into diatoms, diatoms produce 20% of the oxygen that we breathe. So every fifth breath, you can thank a diatom for. They are typically single-celled, though they will form chains. So each individual cell in the chain can function independently, but they will form chains in some species. They are made of silica, and they have this shell structure. So two valves, a top and a bottom valve, and the girdle is the side where they connect. And that whole structure together is called a frustule. There is a huge variety of shapes and sizes in the diatom group. And so this is just one of them. And they even, because we're looking at all of this plankton that is 3D in a two-dimensional way on a microscope, we may see a variety of shapes and sizes within one species just because we're looking at it from different angles. They are primarily reproduced by splitting. So that top valve and bottom valve will come apart and create new adjoining valves. So they get slightly smaller every time they split. And then after a period of time, they will produce gametes and create a one larger cell again and start the process all over. And like I said, they are photosynthetic, so they are producing oxygen with sunlight and various nutrients. In this photo here, we have two cells that look very different. However, this is the same species. This is a Cocinodiscus cell. And this is the top view, the valve view. This is a centric diatom, so it is circular. And then this is a girdle view. It's the side view, so looking directly like that. And so you see a very different view of this species depending on how the cell is sitting on the microscope slide. And so some notable diatom species, these are ones that I find really interesting. Again, Cocinodiscus, this is a different photo of a similar species. So this is the valve view, top view. We see this nice circle there. This is the girdle view. However, what we're seeing is that the cell was large enough that the microscope slide and the cover slip, when they sandwich the sample down, actually are breaking that silica or glass casing that the cell lives in. So these are not normal. This is from cracking open the cell on the microscope slide. These three photos here are all Didylum brightwellii, which I would say is my favorite diatom. It might be really nerdy to say I have a favorite diatom, but here it is. It's my big secret. But it's mostly because it's a really fun word to say, Didylum brightwellii. But it has a triangular shaped valve. So this is the flat end where you see the spine going down, you see the spine coming here. So this end looks like this. So it's got a triangle. And then typically you see these on the microscope with on their side because they are a relatively large cell and they get squished pretty easily. And then this photo, I was really excited to find that you can actually see the valve view and then also the girdle view here as well. So this is girdle view, this is valve view, and this is kind of a nice mix of both. So you can see that it is in fact a triangle on the end. And then Chaetoceros, SPP, because there are so many species of Chaetoceros here in the Puget Sound. These are just 4 different ones. Each of these small little golden spots are the cells. So this is one of the chain forming species, and they are identified somewhat most of the time by their spines and how the spines interact with each other. So you can see this one has really long end spines to the chain, but the center spines are a little bit shorter. That can be an identifying factor. This one, you can see they cross over a little bit. This is a curved chain of Chaetoceros cells. So each one of these is a different species chain forming, but each cell can function on its own. And then moving into dinoflagellates, there are 2 main distinctions in the dinoflagellate group. You have the armored, so that would be here. They've got, I believe, cellulose plates, but that might be incorrect. Plates that hold the cell together. And you can see there's very clear distinctions on these segments. Or you have unarmored or naked dinoflagellates. This would be this one. They're a little bit more flexible in their shape, but they still have a standard shape that they can be in. Most dinoflagellates are solitary, but there are a few that form chains, including one of our toxic species here in Washington. And they are most identifiable from diatoms by their movement. Dinoflagellates have 2 flagella or little tails that will propel them through the water in a spinning motion. They also reproduce by splitting, similar to diatoms, but that's a whole nother talk that we'll go into another day. And these are some of the mixotrophs that I was mentioning before. They can be photosynthetic, they can be grazers, or they can be both in some situations. So they're just a really great diverse group of single-celled organisms that we find in our water. Here are a few different species that I find really interesting. This is Protoparadinium. Here on the side, there are several different species of this as well in our local waters. In this video that I'm about to play, you can see the central flagella going around the side. You'll see it wiggling, and then you'll see the forward motion flagella coming out the bottom here. So if we play this, you can see that movement there. We zoomed through the slide. There's that backwards facing flagella. And the reason this cell is not moving is it was large enough that it was stuck under the cover slip. And then this is Polycricos. I like to think of this one as a honeycomb cell. It also has a flagella that goes backwards so that it can propel itself forwards, but it has several different loops around it for that central flagella. So it just has a really unique look to it. And so I always love finding these under the microscope. These 2 species, Nematodinium armatum and Erythropsidinium, are 2 that are really intriguing to me. Remember that these are single-celled plankton. They do not have organs like we do. They have organelles. Single-celled plankton. This little spot here is an eye spot. It's called an ocelloid, and they are actually able to detect light and concentrate light with it. And it's thought to be used for hunting. So you can see this one is spinning. So that central flagella is kind of pushing it in a circle as it moves around. But this organ here, that was some sort of something flipped by. But this little organ here is used for light sensing and for hunting, which is really incredible for a single-celled organism. This one also has an eye spot right here. You can see it. And this one, rather than using a flagella for movement, it primarily uses this piston. I've only seen this species twice, so I'm really hoping I can catch it on video again sometime in the future. But just really, really cool dinoflagellate species found here in Poverty Bay. All right. And then I'll touch on larger zooplankton. There's too much of this to cover in a 45-minute talk, but most of this is multicellular organisms in a variety of classifications. And some of them will stay small. So we've got our copepods that really only reach like a millimeter or two at the very, very most. But we also see a lot of larval forms of our larger invertebrates that we would find tide pooling. So we'll find larval forms of crabs. We'll find large larval forms of sea urchins and brittle stars. We'll find anemone larvae. We find worm larvae. And so some of those are right here. Sometimes we even just find dividing eggs. If we have a broadcast spawning event in the local water, we'll sometimes just find clumps of 4, 8, 16 eggs. And we don't necessarily know who they belong to all the time, but we do know that they are that some kind of spawning event happened. And sometimes, if we're lucky, we'll start to see the larval forms in the following weeks. In the springtime, we see an abundance of barnacle nauplii. These are larval barnacles. So we think of adult barnacles in a volcano shell, stuck to rocks, stuck to aquarium glass, all of the things. They do start their life as a very mobile little crustacean. They have these little horns that are used to ID them, and they swim around the plankton, eating all the diatoms until they find a spot to sit and become an adult barnacle. Polychaetes are a type of worm. This one I just thought was a really cool photo. I'm not exactly sure what species this becomes, but the polychaetes are in the bristleworm category, so they have all these little spines on them, which is super interesting. Both of these are copepods, so we have a lot of different types of copepods. Harpacticoid copepods are typically found more on the bottom and on surfaces, but there are some species that can be in the water column. And this one is an adult. You can see these are the Coscinodiscus diatoms for size reference, so they're still pretty small. And I know this is an adult because it has an egg mass here. And then this one is an adult Calanus copepod. This one's more of a planktonic species where you're going to find it in the water column. And these big antennae are a good giveaway for that. This is a snail larvae. So this is a veliger, so a stage of the snail development. And it looks just like a little teeny tiny snail shell, and then it's got some cilia that come out of this opening here. Both of these are echinoderm larvae. So we've got our sea urchin larvae here, and our, I believe this is a brittle star larvae. I found this just recently, so I haven't 100% confirmed the ID, but based on what I can find, it looks very much like a brittle star larvae. And these are Pluteus is the stage, but they often eat diatoms. This one you can see some diatom spots inside the gut, which is super interesting. And then there's lots of other types of plankton that we won't have time to go over today, but we've got Dictyocha, which is a photosynthetic species that has this silicon skeleton here. So this is one that has all of its tissues. This is just the skeleton. There are flagellates, which are similar to dinoflagellates, and they have flagella, but they are shaped very differently. And this one in particular is quite a bit smaller. And then we have a wide array of ciliates. So these are just a few that we see regularly in the plankton. So moving on to where is plankton found? The short answer is everywhere. Plankton is going to be everywhere in the water column where there's sunlight because the photosynthesizing species need sunlight in order to grow. And then some of the non-photosynthesizing species may be found a little bit deeper. And a lot of plankton species are part of the largest daily migration on Earth. This is our vertical migration where species will go up and down in the water column every day, depending on the time of day. And then here in the Salish Sea, the sunlit portion of the water column is actually relatively shallow. If you ask any diver, you'll hear that if you get down to about 100 feet, even on a bright sunny day, it's pretty dark already. So there's not a lot of sunlight sunlit portion of the water column here in the Salish Sea, but there is definitely lots of plankton still living here. And in this photo, you can see that some species will congregate at the very surface of the water. This was taken while I was snorkeling just off the dock. We were collecting moon jellies that day, and about 3 or 4 inches of the surface of the water column was just thick with plankton. And most of the species, there was a lot of different species in this, but most of what you see here is Noctiluca. And Noctiluca is a species that we'll be talking about in regards to blooms in a few minutes. So I won't spoil anything there. And our friend Cosinodiscus is also visible in this photo in these little smudges of brown. They get almost large enough to see with the naked eye. So this is what they look like under the microscope. And this is what you might see if they're blooming in the surface waters. And then when is plankton found is another good question. Plankton is found all the time in the water. We always have to have some plankton in the water, but there is definitely a seasonality to it. So during the spring, we get spring bloom of especially diatoms, but other species as well. We have very cold water, but then we start to get more sunlight. So the diatoms start growing like crazy, followed by the zooplankton and then followed by other species. So it really kicks off our ecosystem every year. And this chain here is very simplified, but it is something that we see pretty strongly here in the Puget Sound and in Poverty Bay. We see the diatoms start to show up in mass in March-ish. That's also when diving becomes significantly more difficult with low visibility because of all the plankton in the water. And then the copepods, other zooplankton, start to eat the diatoms in April, May. Then June, July, you'll start to see moon jellies start to increase in number. And then that's followed shortly or coming at the same time as egg yolk jellies. Egg yolk jellies are medusivores, so while they eat a lot of different things, they do primarily eat moon jellies. So this is a very straightforward food chain that we see here in the Sound pretty much every year. And so who is monitoring the plankton is also a big question. And so there are two organizations that I have worked with in the Puget Sound that are on the forefront of the harmful algal bloom groups, one of them being ORHAB. ORHAB is the Olympic Region Harmful Algal Bloom group that monitors the outer coast. So on the map here, you can see they monitor out here. And it was established in June 1999, and I'll just quickly read their mission. It's also on the screen. ORHAB shares knowledge with local communities on the Olympic Peninsula of the Washington State coast, empowering tribal and state managers to make scientifically based decisions about managing and mitigating harmful algal bloom or HAB impacts on coastal fishery resources. And so if you've ever gone clam digging on the outer coast, this is the organization that is saying, yes, it's safe to go clam digging. And then on the inner coast in Puget Sound, Sound Toxins is the primary monitoring group. It was established in 2006 and is currently led by Washington Sea Grant. The overall goal of this cooperative partnership is to establish a cost-effective monitoring program that will be led by state managers, environmental learning centers like MAST, tribal harvesters, and commercial fish and shellfish farmers. The Sound Toxins program aims to provide sufficient warning of HAB events to enable early or selective harvesting of seafoods, therefore minimizing risks to human health and reducing economic losses to Puget Sound fisheries. And so this is the organization that we partner with as a Puget Sound facility. They have samplers all throughout the Puget Sound and are able to get a really good idea of what's blooming every week in the Sound. And so those are the two organizations that are kind of leading the charge with monitoring, but how exactly do we monitor plankton is a question. So we want to take a plankton sample. We use tow nets. So this is a phytoplankton tow net, and they can be of a variety of different sizes of mesh. And so this looks like it's a solid fabric, and that's because the mesh size is 20 microns. So very, very, very small. There are some species that are smaller than that, but on the whole, 20 microns will catch everything that we're looking for. And what we do is we lower this net directly off the end of our dock down into the water until we hit the bottom bottom, and then we pull it straight back up. And so we get a vertical tow that will tell us what is in the entire water column. You can also do a horizontal tow by dragging it along the edge of the dock, but you won't necessarily catch what's down in the deeper water. And so when we're looking at the plankton sample, we are using a light microscope. These will magnify 40 to 400 times. And so we're able to see quite a bit of little small things. We've got our microscope slide and cover slip. So we put a little drop of water onto the microscope slide and then kind of sandwich it with the cover slip, just so everything is nice and thin. We're able to see what might be on the slide. This was a particularly concentrated sample. This was back in the spring. And so there was a lot of plankton to look through on here. And so then you just look through the eyepiece and see what you see. And you can also, with technology is fantastic. You can get adapters for your phone to line up your phone camera with the eyepiece so that you're able to take pictures through the microscope. That is one thing that I do a lot. That's where most of these photos come from, except usually I'm just holding my phone up to the camera lens. But these are really helpful to get videos and really stable images. And then when we're looking at a plankton sample, I love photos and videos for this because especially videos, because you're able to see different angles if you have something that's moving. A lot of species, like we saw with Cossinodiscus, might look very different depending on the angle that we're looking at it. And so you may have one picture and you're like, I know exactly what that is, but you could have a picture of the same thing in a different position and it looks completely different. And so with this video that I have here, you can see some movement. You can see it twisting around. But one thing you can see is that there is a flagella here and there is a flagella here. And so if we watch that again, you can see those moving and it tells you, okay, so this is the dinoflagellate. And then we look at other characteristics. This cell is relatively round. It's got a very high central flagella and this little wing on the side. And so I know that this is a Dinophysis species. But these are just a lot of different ways to identify things. And sometimes identification is impossible without a higher powered microscope. So sometimes you need an electron microscope. However, what we're looking at is typically on the scale that we're able to identify to what we need to with the light microscope. And then if we take a picture and we want to look at it later, and we're looking and we're like, what do we have in this photo? In this photo, diatoms are generally golden brown. Some dinoflagellates are as well, but diatoms have a very solid structure because they have the silica, the glass-based shells. They're typically very regular in their shapes. And so all of these we see are the Didelum brightwellii. These ones are chained up. These ones you can see are actually splitting to form new cells. And then we have some Chytoceras species here. Here we can see their spines and their nice solid chains. Dinoflagellates are typically less consistent in shape, and they're most easy to identify when you have a video because then you can see the movement as well. And so in the next slide we'll see movement. But this here is a dinoflagellate. It doesn't look like a diatom, even though it is the golden brown color. It's got you can see almost a central groove for a flagella, and then it's going to be moving around in the next slide. Ciliates have hairs. So instead of two flagella, they have a variety of little hairs around their body. And so you will see those sometimes. And they're sometimes really, really small, and you will just see something zipping by on the microscope. And sometimes they are big enough that you're able to take pictures of it. And so if we look at a plankton video, diatoms typically don't move. There are a few exceptions where they are able to extrude their internal fluids and kind of slide along the slide. And so there are some that will move a little bit, but usually if your diatom is moving, it is because something else is pushing it. Dinoflagellates do move, and they have a very specific pattern of movement because they have that central flagella. They will spin, and then they have the one that pushes them forward. So they spin and they move forward in this kind of a pattern. This one was trying to swim against the microscope slide, so that wasn't super helpful. But you can see the spinning motion, and then when it turns, you can see the flagella pushing it forward. And so that's one way that we can see, okay, yes, this is definitely a dinoflagellate based on its movement. Ciliates are very fast. I don't have any videos of them in this presentation, but their hairs beat constantly and they look kind of like this. And so you can tell a ciliate from a dinoflagellate based on how many moving parts it has. And so, honestly, why do we care? So, we care about plankton a lot, not just because they're cool, but because they are incredibly important in the ecosystem. We have our primary producers, so especially our diatoms. They use nutrients and sunlight. Some of those nutrients come from us, so human created nutrients or human off-gassed nutrients, and they take that in and produce oxygen. And they are also part of the carbon cycle, so they are really important in that regard as well. Not all blooms are bad. So the spring bloom of the diatoms and the phytoplankton that happens in the springtime is really important. It sets off the ecosystem each year, and so it helps with some of the larval growth throughout the spring and summer. And then we also have the harmful algal blooms that can produce toxins or negatively affect marine life and humans in other ways. So speaking of harmful algal blooms, we do have some species that we are looking for here with sound toxins. This species, Pseudonychia, is a diatom. It is one of the ones that will slide along the cell by extruding its guts. And then we have two dinoflagellate species here. We have Alexandrium, which is a dinoflagellate species that will chain up. Not very many of them do that, but the species does. And then we have a Dinophysis species here with the wing and the high flagella groove there. So Pseudonychia is a species that produces domoic acid and causes amnesiac shellfish poisoning. If you've had a clam dig tide canceled, it's almost always because of this. Not always, but it can definitely be this reasoning. And so Pseudonychia with domoic acid and amnesiac shellfish poisoning, flu-like symptoms plus dizziness, seizures. It can be really, really bad if it's a really big bloom. It can even cause a coma. So definitely not something to play around with and something we want an early warning system for. Alexandrium produces saxitoxin and paralytic shellfish poisoning, or PSP. This also has some neurological symptoms and can result in tingling and numbness within an hour or two of contact. So it is very fast acting. And because it can cause paralysis, it can lead to respiratory events and cause a lot of issues very quickly. Dinophysis produces okadaic acid and a couple others, and it leads to diuretic shellfish poisoning, which is exactly what it sounds like, flu-like symptoms, and not something you want to mess around with. And then there are some species that are kind of up and coming harmful algal species or ones that are causing issues in other areas of the world. Akashiwo sanguinea is not a toxin producer necessarily, but it is associated with birdfish and shellfish kills because it produces so much mucus and causes low oxygen events in the water. So when this blooms, there's really slimy water, just not a good time all the way around. Noctiluca scintillans is also not toxin producing. We get it blooming in the sound pretty regularly. It's if you see tomato soup color in the water, this is one of the species that causes that. And while it's not a toxin producer, it can also bloom so much that it causes low oxygen environments that can cause issues in the food chain. Protoceratium reticulatum is another one that produces yesotoxins, which I believe is kind of more of an up and coming area that's being studied, which is also associated with diuretic shellfish poisoning. So like Dinophysis can cause some issues when it blooms. But what about red tides? You go to the beach and you see this water and you're like, that doesn't look healthy. I don't think I want to interact with that water. So not all red tides are toxic, and not all toxic species and harmful algal blooms produce a color change in the water. So this is a Noctiluca bloom we had here at Mast earlier this year. I believe it was in April or May. And the water was that video that I just showed. You can see in the background of this photo that there is very tomato soupy colored water. And this beaker, I just scooped up a sample. This is not concentrated in any way. Um, and so Noctiluca does bloom here. We haven't had it associated, to my knowledge, with any mortalities of marine life or anything. But it is something that we keep an eye on because it is a very much blooming species. And something really interesting about Noctiluca is that it is not photosynthetic. It does not um, if it does not produce oxygen or have chloroplasts or anything, but it does eat other diatom species and other plankton species by engulfing them. So this is a Noctiluca cell here that you can see it's eaten 1, 2, 3, 4, 5 different chains of diatoms. And you can see these ones have also eaten some chains of diatoms. And what they do is they basically engulf the chain, bring it in, take what they need from it, and then they spit it back out. And actually, I was lucky enough to get this on video in a time lapse. And so you can see the flagella moving around. It's got this groove here. And there are at least 2 different species currently inside this Noctiluca cell. And so if we watch in just one moment, it rolls over and spits out the chain of cells that it had engulfed previously. So just a super interesting technique for eating, something you may not expect from an eyeball looking dinoflagellate species here in the Sound. All right. And then another species that will change the water color is Tripos. Tripos is another dinoflagellate species. It is an armored dinoflagellate species, so it is very sharp. It's got the nice shell to it. And this bloomed last summer, and we came down to the Mass Center one morning, and the water was rusty red, just like this. And it is very sharp. It is linked to harmful algal blooms in Eastern Asia. From what I could see, because it is so sharp, if it got into the gill tissues in high enough quantities, it could cause issues with shellfish or fish. And so it's not considered a toxic species here in Washington, but it is one that will bloom and cause very rusty red colors. And so, woohoo! Hopefully, you agree with the statement that I now understand how phytoplankton monitoring is done, but how does that affect me? How does all of this really cool research and data collection influence just everybody? So Washington shellfish eat plankton, and Washington's people eat shellfish. I couldn't find specific data on how many shellfish licenses are purchased from Washington each year, but over 800,000 fishing licenses are purchased in Washington state each year. And also, unless you're growing your oysters or your shellfish in a closed land-based aquarium system and creating your own seawater, most farmed shellfish, like oysters, eat the phytoplankton that are out in nature. And so monitoring the shellfish monitoring the plankton allows us to have an idea of what's going into our shellfish that is then either eaten by our local people or sold as an industry. Washington does lead the nation in producing farmed shellfish, so this is really important that we know about all of this, and produces $107 million worth of shellfish each year. Washington Department of Fish and Wildlife is the organization that monitors razor clam tides. So every time there's a clam dig weekend or tides, that's always being double checked by the plankton monitors on the outer coast. And this I thought was a particularly interesting find that Washington Department of Fish and Wildlife recently shared on Facebook. A dinoflagellate tripos. So if we go back, this is tripos, or one of the tripos species. actually produce this rusty red color in the razor clams and the oysters. So it is something that even if it's not a toxic species that is happening, that we're able to monitor and understand when something happens like this. Wow, that's really cool. So, what can I do? If you want to get involved, if you're local to the Mass Center, we are always looking for volunteers with our Sound Toxins partnership project. We take weekly phytoplankton samples. This was just this last week on Halloween. And so we take weekly phytoplankton samples, and then we spend some time on the microscopes getting to know the different plankton types, both harmful and just local. No experience is necessary. You can come hang out, learn how to use a microscope, talk about plankton for another couple hours with me. And so we're always looking for new people to do that. And also, if you're not interested in phytoplankton sampling, but you want to learn a little bit more about the shellfish or clam digging, look up Washington Department of Fish and Wildlife. They post regular updates to what beaches are open, what ones are closed, kind of how that's done, and why it's regulated the way it is. And if you're in Washington or another coastal state, check with your local fish and wildlife organization. They will be monitoring likely a very similar way. So thanks for joining. If you have any questions, please leave them in the chat. Or if you have any questions for me, email me at my email here that's on the screen. And thanks for joining.
All right, awesome. I've got a few questions here about some of the photos that you were showing, to about just plankton in general, because even though you work in the marine field and you understand plankton, it doesn't always fit into those nice, tidy, neat little boxes. So my first question is, you were talking about plankton and categorizing them, but there's always like the exception to the rule, right? Can you talk more about the non-animal, non-plants that are out there? They're not in the animalia kingdom, so like, what are they?
So yes, so Kingdom Protista is a really interesting, just like catch-all group. Animals are there's certain cell types that are considered an animal cell type. So you have sponges, which are nothing like people, but they have that specific cell type. So they are considered animals. Plants, same thing, very specific cell type. Fungi, again, something I don't know much about, but it's own thing. It's in a kingdom, and then Protista is just kind of everything else. So a lot of seaweeds are in Protista because even though they're very plant-like, their cellular structure is different, and so that's how a lot of the phytoplankton. It's kind of in there. It wouldn't surprise me if eventually it's completely overhauled and is like, okay, here's four more kingdoms and they're their own thing. But yeah, it kind of is a catch-all group for everything that doesn't really fit anywhere else.
Okay, so you were talking about dinoflagellates and being able to move, but we're single cellular, but we're also starting to grow eye spots. Is that where then, as these plankton start to evolve and our better understanding of genetics and barcoding, that we might start to see them broken out into smaller and smaller groups, or changing from one group to another group?
Yeah, I would say so the way that science in general, but definitely plankton, everything originally was based on what it looked like. So if this animal looked like this animal, they must be closely related. And so with genetics, we're sometimes finding that yes, we were correct, or no, we were completely off base. And so with that sort of barcoding and genetics, that may be something that will help kind of fill in the gaps of our knowledge of what is related to what in the plankton.
Okay, so let's talk about the odd one with the eye spot. As an animal who, or non-animal, we're in that gray territory. As a being who can't swim against a current, so doesn't really have the option to really move into that certain direction, why is that like a evolution of an eye spot? Like, why? Like if I can see it but I can't move towards it, why does it matter, right?
So when you get down to that scale of plankton, you have a really fun physics concept called Reynolds number. So for us, water, we can move through it pretty easily. We're able to fight against it, I guess. But on that size scale for plankton moving through the water, it's more like us moving through like maple syrup or oobleck. So it's a very different environment than we're experiencing, which is one of the reasons why it's so much fun to study because it's so different. But so they are able to move somewhat, but it's just a very different type of movement. I was actually looking up some information on the one with the piston just recently, and it the way that it contracts its and releases its piston, it's a way that it does push it forward. I believe it one speed is faster than the other. So again, I got to look into it more because it's so bizarre. But it just allows it to it's a different way of moving than we're used to. So
Okay, so most science is looking at, you know, these weird evolutionary traits and kind of like turning them towards science and using them for building mechanisms and making our life easier. Do you see us as humans trying to mimic that said like piston mechanism in order to like advance machinery, or using that sort of eye spot sight like or lens for advancing humans, or you know, things like that? Do you see any sci-fi movie novels things coming out of this weird world of plankton?
I mean, I think there's always opportunities for to learn new things and to kind of expand what we can do and use, little bits of this and that and make something really cool that's brand new. What's really interesting about the iSpot is that it is very much like a camera that it concentrates the light. And so it does things that you wouldn't expect a single-celled organism to be able to do, and things that we do use on a daily basis now. And so some of those traits we're using, and maybe we can learn some things from the plankton to move forward.
Awesome. So you were talking at the beginning, we have these different groups of the plankton. We're talking about phytoplankton and zooplankton, phytoplankton being more plant-like, zooplankton being more animal-like. And then you were talking about how some plankton start off small and then grow up into these bigger things. Can you talk more about, I guess, what does start off as plankton or has like a portion of their life as plankton that we see and that we don't think as plankton? Because I know jellies, most people see jellies and are like, oh yeah, that's not plankton. But some people are now starting to catch on that, yes. Are there other things that would be like, oh my gosh, I had no idea that's plankton, or like no idea it started off as plankton?
So yeah, a lot of things start off as plankton, especially in the marine world. Anything that broadcasts spawns, so releases gametes into the water, pretty much starts off as some type of plankton. Sometimes we see it as early as, 4 cells in a clump. That's gonna be something, but it is in the plankton at that stage. Pretty much every echinoderm, except for a few sea star species, I believe, start as plankton. So sea stars will release their gametes. They'll have a couple different planktonic microscopic stages. Urchins, as we saw, brittle stars, as we saw, cucumbers will go through that as well. Usually they have a few different transformations before they settle to the seafloor and become the adult species that we know and love. And so like then you have your crustaceans, so your crabs, you have your barnacles, you've got shrimp, you've got hermit crabs. All of those have multiple planktonic stages before they settle to the bottom as an adult. Even fish will have planktonic stages sometimes, whether they are, a few days or a few months. Recently, we've grown gruntsculpin here. They have a very extended planktonic stage. We found it was about 5 months. So yeah, a lot of species in the marine world have a planktonic stage.
So I have been told that mola mola are plankton, and that we have mola mola in Puget Sound. Can you talk about how a mola mola, which is a warm water fish, could potentially get into Puget Sound? and being a
Yes.
plankton, even though it's a super big fish.
Yes. So mola mola, I actually looked that up specifically before I did this because I was like, yeah, I want to talk about the largest, bony fish that's plankton. And apparently there is some research that they may be able to swim against currents, but they aren't very good at swimming against currents because, like you said, we do see them here in the sound. Occasionally, I know that one was found in Redondo not that long ago, here in Poverty Bay. What happens is they get pushed on those warm water currents that get pushed up into Washington occasionally. And if it's a strong enough current, they aren't able to swim against it. And they just end up where they end up and in bizarre places like Poverty Bay here.
I mean, I would also want to just, ride along on a warm current for
Yeah.
my life.
Until
So,
you get out of that warm current and then you're like, oh, this is cold.
so with the currents, you're talking about like these tomato soup tides or these rust tides, where the plankton are blooming. So this is kind of like a two-part question. What are the environmental factors that like result in a bloom, or like how do we predict that there might be a bloom happening? And then when those blooms do happen, like a tomato soup or a rust tide, how do we know that it's safe to interact with that water? Because like that beaker, you went out there and scooped up that beaker. How do you know that like it's okay to touch that water or be near that water during that time? So again, first question, what are the factors that lead to a larger bloom like that? And then second, how do we know if that bloom is okay to either swim in or, touch?
Yeah, so there are a lot of different reasons that will, or a lot of different factors that will lead to a bloom happening. Sometimes it's nutrient based. Pseudonychia will bloom if there is a sudden input of iron in the water, because they're really good at taking up that iron, whereas other species are a little bit slower. So sometimes it's nutrient wise, we can tell that it's going to bloom. In the springtime with the spring bloom, it's really cold water and the addition of sunlight. You start to see just this bloom of all sorts of plankton species, because they're just like, got to use that sunlight, got to use the cold water that's very oxygenated. And so environmental factors, sometimes currents will change blooms, mixing in the water column. So if you have a big storm, and then you have a couple of days of sunlight, you're going to have really nutrient rich, oxygen rich cold water and sunlight. So you're going to have all the right things for the diatoms to start blooming. So there's a few things. There's also things that we can't predict, especially with climate change, with everything becoming less predictable. That can be a challenge that we're facing now, to see, okay, why is this blooming? And then you also have certain species that will eat certain species. So if you see a bloom of one and you know that there's a species that's the preferred prey of, you might expect to see the predator following the prey. And then how do we know it's safe to touch? Most of the time, if you see something like that, some organization in the world has been alerted that, oh no, the water's red. And so sampling will be done. So the best way to know if it's safe is to see if sampling's been done, Google something like that. And so yeah.
Okay. So we were talking about tomato soup tide, and it's caused by Noctiluca, which Noctiluca meaning nightlight, so bioluminescent plankton. And there's a few different bioluminescent plankton here in Puget Sound. Is there a place, I know sound toxins we're monitoring for everything, but is there a place that's monitoring specifically for bioluminescence so that people, when it does happen, we can go out and enjoy it because it's such a magical thing to see. And it really needs to be like a bigger bloom in order to like, really enjoy it instead of just having a few sparkles here and there. Do you know of any organizations that are like tracking that, or any place that the public can go if they're looking for bioluminescence in Puget Sound?
So I don't, off the top of my head, I don't know of any specific organizations that are monitoring for bioluminescence, but I know that it is something that just everybody looks for. So I do know there are a couple pretty sizable Facebook groups that will like report bioluminescence here and there, especially in the summertime is when we tend to see the bioluminescence. There are also some outdoor centers and places that do bioluminescent kayak tours. So they would be someone that you could probably contact and say, "Hey, are you sampling? Do you know where you get your reports for this?" And so Facebook though is great. There's a lot of different bioluminescent groups that they may not be sampling, but they're out there looking for the plankton.
No, we've definitely posted on our social media about bioluminescence and plankton before. So with plankton being pushed around with the currents, so say we have bioluminescence at Redondo one night, would then people be able to follow that current north or south depending on where the Noctiluca ended up, or the other bioluminescent plankton? Would they be able to follow that to go to know like the next beach is going to be like Lincoln Park or Alki, or is it kind of like just dispersed? It goes where it goes. Is there a trend or a pattern?
So sometimes if you have really established currents in your area, you may be able to predict like, oh yeah, it's gonna go to the beach just north of us, or it's gonna go just south of us. But also it can depend on just like the way that your coastline is set up. So I not here, but over on the other side of the sound by Poulsbo and Bremerton, there's two different inlets of water. And I know that Sound Toxin often samples both of them separately because even though Bremerton and Poulsbo are only about a half an hour drive apart, the waters are very different and they can have two different things blooming because they're not super connected. They connect in the Puget Sound, but they don't connect deeper into the bay. And so even small distances can have very different plankton populations. So sometimes you can tell, sometimes it's anybody's guess.
So I don't want to freak people out, but we were talking about all the living things inside the water, because you might look over at the water right now at Redondo and say, "Oh, that's really clean, really clear." But as you were saying, even in clean, clear water could potentially be these plankton. So I know if you take a gulp of water, you jump in the water, you figure to close your mouth, or you inhale water. That's not something to be concerned. Can you talk more about like the quantity in which you need to either consume or how it's consumed that it's actually a harmful algal bloom to humans?
Yes. So, with harmful algal blooms, we, with phytoplankton sampling, are the early warning system. So most of what we're concerned with is once it gets into the shellfish. So with sound toxins, if we take a sample and we look under the microscope and we see there is a lot of Alexandrium blooming in here, which is the paralytic shellfish poisoning producing species. So what we do is we contact sound toxins and say, hey, we have a big bloom here. You may want to alert Department of Health. And so they alert Department of Health, who then puts out mussels or some other shellfish to actually test how the shellfish are taking up the water. It's not necessarily the water itself that's the problem. It's how much is being ingested and accumulated inside the shellfish. And so then they'll test the shellfish and say, okay, yes, we're going to close this beach, and this beach until we test clean. So it's not. If it's in the water, yeah, it's not a good thing, but it is not something that's necessarily going to hurt you if you ingest some. We also concentrate our plankton samples that we're looking at. So if we see, 50 cells in our slide, then there's a lot out there, but it doesn't mean there's 50 cells in your gulp of water that you're taking. And so it's all based on different thresholds that the scientists have established, so that if we hit that many cells per liter, then that's like, okay, maybe we need to start testing the shellfish. So it's really like an early warning system for the shellfish, not that this is all in the water and it's going to kill me kind of a thing.
Okay, so there's lead up time. So
Yes.
okay,
Yeah.
anybody watching who might have been freaked out by all the little things in the water, there you go. It's the amount of concentrated plankton that
Yes.
does it. So I know we're getting here on time. So you were talking towards the end about volunteering and not needing any experience. So can you talk more about the volunteer opportunities here at MAST with the harmful algal bloom monitoring, and what they do, what a day in the life of a monitor would be?
Yeah, so here at MAST, we have weekly plankton sampling on Thursdays from 12 to 2. So once you're in our volunteer system, you can come and on your first day, your first couple days, you're either sampling with me or Ariel or with one of our more experienced volunteers. And you'll go out and take a plankton tow and then look at our data sheet, record everything that needs to be recorded, our weather, tides, temperatures, all the things. And then you come back inside and we sit down on the microscopes. We show you how to use the microscope if you've never used it before, and then how to prepare the slide to look at the plankton. And then we spend usually an hour, hour and a half on the microscopes. The first day, I always really encourage people to draw what they see, because if you draw what you see, you're looking at the different characteristics of them, and so it gives you an idea of kind of what to look for when you're drawing. You're like, okay, so it's got a circle and it's got, this little thing that pokes off the side. And then it also helps you describe it when you need to ask me, like, hey, what is this? I'm like, well, what does it look like? And then you can kind of work through the characteristics. And then once you've done it a couple times, you'll start to learn the names. And then once you feel comfortable, you may be the official data taker. We always have one official data taker of the day, whether it's one of us or one of our experienced volunteers. And that means you're counting the harmful cells that you're seeing. So it's just a really great time spending microscope, looking for stuff.
So you're talking about plankton seasonally, and I know during the wintertime we don't get much plankton. There's still plankton out there, but it's not
Mhm.
as much as you would in the summertime blooms. So would it be better, or if somebody really wants to learn and take their time learning before they have a lot of plankton to look at, would you suggest them coming during the off season or like the more winter months?
I definitely would. Right around right now, actually, we're seeing a really good diversity of plankton, but not in overwhelming amounts. So it's not like there's 1,000 cells on your slide. There's maybe 200. So it's a little bit less overwhelming. It's still overwhelming, I think, for first people. So it's just like we take it very slow. But it's less overwhelming now than in March when you have brown water and you've only done a meter of the water column. So
Perfect. Well, we are officially out of time. I want to say thank you, Emily, for
Mhm.
talking about plankton. You clearly love plankton with all of the photos and
Absolutely.
stuff that you have. So if anybody does want to be a part of our harmful algal bloom monitoring project with sound toxins here at the Mass Center Aquarium, you can find out more information about our co-science teams on our website, mass.highline.edu. You can also turn in a volunteer application via our website so that you can start volunteering here on Thursdays from noon to 2, looking at plankton. And if you're interested in the zooplankton side, we also have our light trap monitoring program also, which happens on Mondays, Wednesdays, Thursdays, and Saturdays from 9:30 to 11:30. So if you want to come and hang out and talk nerdy about plankton, we're always down. But if you do have any questions, feel free to reach out to Emily or the Mass. And I know we'd be happy to answer and talk about plankton. So,
Absolutely.
all right. Thanks.
Awesome.